Hi everyone. So in this video we're going to talk about cell adhesions. So as you can see here, this part is always called the apical surface, and it has microvilli here because, let's say for example, this is an epithelial cell, and this is right here is the lumen, the gut lumen in the intestine. So this is the lumen, this is what you eat, and it's here as lumen, right? So in order to increase the absorption surface, there has to be microvilli, where it increases the surface area for the nutrients to bind in and get absorbed. So it's getting absorbed, right? And it goes through the cell, and what's really needed is gonna be having specific transporters, and will be secreted in the blood. But before we go into the blood, there is interstitial space, and this interstitial space is extracellular matrix, and it's basically in this part here. So for the extracellular matrix, it's like a lot of um, component are all in together. So the component of the extracellular matrix is basically collagen. We're having collagen. We're having fibronectin. We're having fibrinogen. We're having elastic fibers, and there are actually a lot more. So all of them, they are actually existing in the state of GAGs, or I mean proteoglycan. So basically, proteoglycan is a polypeptide attached to GAGs. So it's polypeptide attached to GAGs. So what is GAG? GAGs is basically glucose amino glycan. So it's basically, it has two subunits. One is amino sugar, and the other one is acidic sugar. Both of them are combined. These are the subunit of the GAGs. And attached to this is polypeptide. So most likely, like collagen, fibrinogen, elastic fibers, all of these are actually existing in the proteoglycan phase. So except for one of them, and it's called hyaluronic acid. And this one is really so important. So it doesn't exist as a proteoglycan. It exists as just GAG. So this one is really so important for the synovial fluid in the joint. So it acts as a shock absorbent. Okay, so all of these things are actually here. So they are just creating networks together here, and it forms a really tight kind of network, so that it's really strong, and it could actually resist any kind of stress or anything like that. So this is the extracellular matrix, and what does it what is the component of it? And basically the whole thing here is the extracellular matrix, okay, combined with the interstitial fluid. Both of them, so this part right here, it will be the interstitial fluid, and this is the extracellular matrix. Both of them are going to be the extracellular fluid. Okay, so let's talk about what are the cell adhesions. So we have three we have three types of cell adhesions. We're having one, anchoring junctions, two, um, occluding junctions, occluding junctions, and lastly we have communication junctions. Okay, so for the anchoring junctions, um, there are four components of it. We're having adhering junction. So what is adhering junction? It's basically this one in yellow. So the cytoskeleton partner of this would be actin. So it binds to actin. As you can see here, this is actin, and it's falling around to the other cell. Its job is to, from the name of the type of the junction, anchoring. So it actually it's cell to cell anchoring. Okay, so this is the first type. The second type, we're having um, desmosomes, and this is the one in blue here. Desmosomes has a different type of cytoskeleton partner, which is intermediate filament, which is in black. This is the intermediate filament, and the job of it also is to anchor. It's making anchoring parts in the cell. We're also having this one here in red. This is semi-desmosomes. So semi-desmosomes also have intermediate filaments, and it has integrins. So it binds to the basement. So this here is the basement membrane. This right here is the basement membrane. And the main component of the basement membrane is laminin. So laminin is different from lamina. Lamina is only exclusively in the nucleus. It's an intermediate also for lamina. So it attaches the cell itself via integrin with the basement membrane. Okay. And lastly, for this one, we're having focal contact. And focal contact is pretty much like semi desmosomes, but it's only different that the cytoskeletal partner would be actin. It's not intermediate filament. Okay, so this is for the anchoring junctions. For the occluding junction, we have only one. So we're having only one type, which is the tight junctions. And the tight junctions, it's basically here on the top, right underneath the apical surface. And the reason why it's right underneath the apical surface, so it creates sealing parts. So it seals both of the cells together so tightly so that nothing from the lumen gets in by mistake. So let's say like toxins or unwanted substances or anything like that could actually leak through the cells, which is unneeded. And for communication, we're having gap junctions. And gap junctions, its main thing is cell-to-cell communication, the transferring of molecules from one cell to another. It's not for anchoring the cells. It's just there for cell-to-cell communication. And then we talked about cell adhesion molecules. So these are the actual molecules that all of these particles that I talked about is using. So for example, adhesion junction, it uses the cadherin as the primarily molecule of adhesion. So we're having four types here. We're having four types here. So we're having cadherin, we're having immunoglobulin superfamily, and we're having integrin and selectin. So for cadherin, what it does, it binds to catenine beta. And this is basically the most common affected part in cancer. So whenever there is cancer, most likely they would find out that catenine beta won't be able to be degraded after the after whatever like usage of this specific cell adhesion molecule. So instead of being degraded, it keeps escaping from proteasome, and it keeps the cells to proliferate. So, and this is why the cells keeps dividing over and over. And it's also very important for cell to cell adhesion. Yeah, and there are two types. There are homophilic CAMs, homophilic, and there is heterophilic. So the difference between them, so for the homophilic, it's basically, most likely it's a cell-to-cell adhesion. Okay, so it's cell-to-cell adhesion. For heterophilic, it could be cell-to-cell, or also it could be cell to the extracellular matrix. Okay, and the difference between them is that a CAM, which is cell adhesion molecules, will bind to the same type of it for the homophilic, but heterophilic will bind to another type of cell adhesion molecules. Okay, so this is the main difference between them. For cadherin, it's usually homophilic, and also it's a calcium dependent. So this one is a calcium dependent. For immunoglobulin superfamily, it could be homo and or heterophilic, and it's also cell to cell most likely, but it's most likely cell to cell. Okay, examples of it is ICAM and VCAM, and the partner, the cytoskeletal partner is actin. Integrin is actin and intermediate filament. And for selectin, lastly, it's actin. Depends on actin. Okay, so for integrin, this one is the most common one if you're talking about cell to extracellular matrix adhesion. Okay, and as I mentioned, the cytoskeletal is actin and intermediate filament. For selectin, it's unlike the other CAMs, it's unlike the other cell adhesion molecules, because this one is found in the blood. For these three, you could actually find them within the cell, right? Could be cytoplasmic, or could be for in the extracellular matrix. But for this one, you actually find it in the blood. And if you find it in the blood, that means it actually helps with the initiation adhesion of leukocytes and platelets to endothelial cells during inflammation and homeostasis. So it helps with the inflammation response, and also blood clotting. Okay, so it helps in blood clotting, the platelets thing. And also it helps with the inflammatory responses. That's it.